Hello Pirates, today we are going to be extracting DNA from strawberry cells. So the ingredients that we need to do this is we are using a strawberry and a plastic bag. So there's materials number one. We have a test tube rack that has a couple of test tubes. We have a funnel, I don't know if you can see the funnel. So there is a funnel. There's cheesecloth, which is kind of like a gauzy, meshy kind of cloth that's used to squeeze cheese curds out, but we're going to separate strawberry juice from that. And then we have a DNA extraction buffer. I have some of that extraction buffer already measured out in this container here in the beaker. And then I also have some cold ethanol. So over here I also have some cold ethanol. I keep the ethanol cold in a flask in an ice bath. So what we're going to be doing is we are going to be extracting the DNA out of this strawberry. And this strawberry is a plant cell, and plant cells are eukaryotic cells. So in a plant cell, there are cell walls on the outside, a cell membrane, a nucleus, and then the DNA. So eukaryotic cells have the DNA stored inside that nucleus that's made of a nuclear membrane. And then we have the cell membrane and then the cell wall on the outside. So in order to get the DNA out, the DNA, because this is eukaryotic cells, they're plant cells, it's going to be stored in the nucleus. So we are going to need to first break down the cell walls of the plant cells. So the way that we're going to do that is just by squishing and smashing them. Okay, so we're going to squish and smash the cells, and typically I usually tell students to smash the cells for a couple of minutes. So for about 2 minutes, we just smash up the cells and just kind of break down the cell walls between the strawberry cells. So 2 minutes of smashing, and I'm going to do this for just a little bit less here. I'm not like the prepared chef that already has the one set up, which maybe next time, but you just want to squish up the strawberries for about 2 minutes. So I'm just going to kind of get this here so I get all the strawberry goo and juice down here at the bottom. All right, so once we have our strawberry smashed material, we need to add the extraction buffer. Okay, so the extraction buffer is a mixture of salt, soap, and water. And I like to use a dish detergent, like the kind of liquid dish detergent that you would put into your dishwasher. And you could use shampoo, you could use dish soap, all of them work. And what this is going to do is the cell membranes inside of the cell walls are made of lipids, phospholipids, and the soap is also made of lipids. So we're just going to mix the soap here mix the soap together with those cells. So I'm just going to squish and use that soapy solution to dissolve the cell membranes inside of the strawberries. Okay, so I'm dissolving the cell membranes as we mix the soap together. And this is kind of what happens when you wash your hands. You've got oils on your hands and bacteria, and when you wash your hands, you are dissolving the lipids and smashing when you're washing the bacteria and dissolving away all those cell membranes. Oops, all right, so here we go. We got the soapy solution. And this is actually how they discovered DNA extraction. It was on an accident. There was a doctor scrubbing bandages, and he was washing the bandages with soap and salt. And he ended up extracting DNA. And then people wondered, what is this? Genetic DNA. All right, well, they just knew it was nucleic acid. They didn't know what it was for at the time. All right, so here we have our soapy solution. So now we have the soapy strawberry liquid. Now what we want to do is we want to filter it out. So we have these solid particles that have kind of stuck to the salt crystals that were in the solution. And now we have this liquid that is strawberry liquid that needs to be filtered out. So I have my filtration apparatus set up. I've got my cheesecloth, I've got my funnel, and my test tube. And what I'm going to do is I'm just going to strain the liquid into the test tube. So So here we go. All right. All right, and I'm just gonna wait for the juices to kind of drip into my test tube. And I'm just gonna give it a little help here. I kind of squish it at the top. Oops, a little came out the end. Ooh, I don't want that. All right, so you guys can see the strawberry juice is dripping into my test tube. And then up at the top, I'm trapping my solids. So the solids are getting stuck at the top, and that's gonna be the other cellular components. So what's in my strawberry juice here is going to be the water, sugar that was in the strawberry, and the DNA. All right, that should be good. All right, so here we have our strawberry juice. And so our final step, so we have the juice. So it's just a mixture of the juice from the strawberry, that soapy, salty solution, and then inside of this is the DNA. But right now, the DNA is within the solution. So the DNA is mixed in the water. And you guys can kind of think like water is polar, and the DNA is mixed in the solution. So DNA must also be polar. All right, so what we want to do is we want to precipitate the DNA out of the solution. So we're going to put alcohol, and we're going to use ethanol, or you could use isopropyl alcohol. We're going to use 5 milliliters, or just shy of 6, I think. And we're just going to add this to the liquid. So there we go. All right, now when I add this to the solution, I'm just going to kind of mix it around here a little bit. We can see that this white stringy stuff has come out of the solution, and it looks kind of like long fibers, kind of like spider webs that are in there. And these long white fibers that are in here right now, that's the DNA. So that's the DNA from the strawberry. Now, strawberries are decaploid, which means they have sometimes 10 copies of DNA, and sometimes they have more than that because they alternate their reproductive strategies between asexual and sexual reproduction. And as a result of that, you could increase the number of chromosomes in the cells. So they're not always ensuring that the offspring are going to be diploid. And so what we're looking at here is the strawberry is going to have a lot of DNA, lots of copies of their chromosomes, and that's why we can see so much DNA coming out of that strawberry. So all that white stuff in there, that is the DNA from the strawberry. So that's DNA, the instructions to make a living thing, and in this case, they contain the instructions to make a strawberry plant and strawberries and make all the proteins and the structures needed for a strawberry plant to function. All right, so what you want to do is finish up your lab analysis and conclusion questions and enjoy looking at your strawberry DNA. And like I said, you can do this at home with garden peas, bananas, kiwis, lots of different kinds of fruit. Again, just kind of creating extraction buffers and salt. All right, let me know if you guys have any questions. Have a wonderful day.